Greetings to all the learners. Today's topic is horizontal gene transfer methods in bacteria. We know that bacteria are unicellular organisms that lack a nuclear membrane, and most of the bacterial genomes consist of a circular chromosome that contain a single DNA molecule with several million base pairs. And we have E. coli as an example, which has nearly 4.6 million base pairs in its chromosomal DNA. Similarly, the bacteria also contain extra chromosomal DNA, what we call it as plasmids, which are also small circular DNA molecules. Here we have episomes, which are also plasmids that are capable of freely replicating, and they have the ability to integrate into the bacterial chromosome, which we call it as F fertility factor or F factor, which we can see in the E. coli, which also belongs to the episome category that controls mating and gene exchange between E. coli cells. Talking about the gene transfer in bacteria, it can be done in two methods, that is vertical and horizontal gene transfer methods. Vertical gene transfer in bacteria refers to the transmission of genetic information from a parent cell to its offspring during cell division, specifically during binary fission. And this horizontal gene transfer involves three different methods, what is called as conjugation, transformation, and transduction. Conjugation is one method in which there is a direct contact between the donor and the recipient bacteria. The donor bacteria is one which contains a conjugative plasmid, what is called as the fertility plasmid or sex plasmid or F factor, and it is denoted by the letter F plus, whereas the recipient bacteria do not contain this conjugative plasmid and it is denoted by the letter F minus. Conjugation is encoded by plasmids or transposons. A conjugative plasmid is self-transmissible in that it possesses all the necessary genes for that plasmid to transmit itself to another bacterium by conjugation. Conjugation genes, or known as STRA genes, enable the bacterium to form a mating pair with another organism. ORIT sequences, which is called as origin of transfer, determine where on the plasmid DNA transfer is initiated by serving as the replication start site, where DNA replication enzymes will nick the DNA to initiate DNA replication and transfer. In addition, mobilizable plasmids that lack the TRA genes for self-transmissibility but possesses the ORIT sequences for initiation of DNA transfer may also be transferred by conjugation if the bacterium containing them also possesses a conjugative plasmid. The TRA genes of the conjugative plasmid enable a mating pair to form, while the ORIT of the mobilizable plasmid enable the DNA to move through the conjugative bridge. Transposons, which is also called as the jumping genes, are small pieces of DNA that encode enzymes that enable the transposon to move from one DNA location to another, either on the same molecule of DNA or on a different molecule. Transposons may be found as a part of bacterium's chromosome, conjugative transposons, or in plasmids, and are usually between 1 and 12 genes long. A transposon contains a number of genes such as those coding for antibiotic resistance or other traits, flanked at both ends by insertion sequences coding for an enzyme called transpoase. Transpoase is the enzyme that catalyzes the cutting and resealing of the DNA during transposition. So here is the general mechanism of conjugation, where we can see in the gram-negative bacteria, the first step is the formation of the conjugation pilus on the donor bacterium, which binds to the recipient bacterium which lacks the conjugation pilus. After the formation of the conjugation pilus, this helps in retracting or pulling the bacteria together. A series of membrane proteins coded for by the conjugative plasmid then forms a bridge and an opening between the two bacteria, which is now called a mating pair. Using the rolling circle model of DNA replication, a nuclease breaks one strand of the plasmid DNA at the origin of transfer site, that is ORIT, of the plasmid. The nucleus also has helicase activity and unwinds the strand that is going to be transferred. The nicked plasmid strand enters the recipient bacterium. The other strand remains behind in the donor cell. Both the donor and the recipient plasmid strands then make a complementary copy of themselves. Both bacteria now possesses the conjugative plasmid and can make a conjugation pilus. The next method of gene transfer in bacteria is transformation, where pieces of DNA released from donor bacteria are taken up directly from the extracellular environment by recipient bacteria. Recombination occurs between single molecules of transforming DNA and the chromosomes of recipient bacteria. The ability of bacteria to take up extracellular DNA and to become transformed, called competence, varies with the physiological state of the bacteria. Many bacteria that are not usually competent can be made to take up by DNA by laboratory manipulations such as calcium shock or exposure to a high voltage electrical pulse, which is called as electroporation technique. The last method of gene transfer to explain is transduction, which involves the transfer of DNA fragment from one bacterium to another by a bacteriophage. A bacteriophage adsorbs to a susceptible bacterium. The bacteriophage genome enters the bacterium. The genome directs the bacterium's metabolic machinery to manufacture bacteriophage components and enzymes. Bacteriophage-coded enzymes will also break up the bacterial chromosome. A bacteriophage capsid mistakenly assembles around either a fragment of the donor bacterium's chromosome or around a plasmid instead of around a phage genome. And this bacteriophages, when released as the bacterium is lysed, they carry the donor's bacterium's DNA adsorbed to a recipient bacterium, and the bacteriophages inserts the donor's bacterium's DNA to the recipient bacterium. This is how the gene of a bacterium can be transferred to other bacterium. And this homologous recombination occurs and the donor bacterium's DNA is exchanged for some of the recipient's DNA. So these are the three different horizontal gene transfer methods in bacteria. Thank you all.